Okay, so Molecular Targets platform. It was born out of the 20 Research to Accelerate Act, the RACE Act for children. And the idea between the RACE Act was to make sure that any new drug that was intended for the treatment of an adult cancer, where the method of action is directed at a molecular target that could be determined to be substantially relevant to the growth progression of a pediatric cancer, would require a limited clinical trial designed to help meaningful data regarding dosing, safety, and preliminary efficacy to inform potential pediatric labeling. So essentially, pharma companies have to test if they have a particular target for a particular compound they're working on in adult cancers. If it's on the FDA's relevant molecular target list, they have to look into a substantial trial in children to check for all of these things listed here: dosing, safety, and preliminary efficacy. So let's see here. So the FDA's relevant molecular target list is born out of an earlier work that they did with experts. It is a list of about 500 different targets, gene targets that come from different families of genes that have been recognized by the cancer research community, pediatric cancer research community, that is relevant for the treatment of these cancers. They also have a list of non-relevant molecular targets which warrant waiver from required evaluation. So these are targets that are not required to be studied if they are in adult tumors or in adult studies. But these particular targets are all relevant for pediatric studies. So the idea behind the Molecular Targets Project was that we were going to bring together a federated pediatric cancer data resource to actually support this Race for Children Act. So we're going to be taking something called the Open Targets Portal, and you can go search for that online if you're interested. It's coming out of the Wellcome Trust. They've originally developed it. It's a great team over there. They've actually built, with a partnership with other pharma companies, they've built a targeting platform that brings in a lot of diverse data sets to try to identify whether a target is, something that you want to address in a particular tumor, or well, not just tumors, but any disease. And our team at CHOP have taken the Open Targets Portal in collaboration with the Frederick National Labs at NCI, and we're adapting the web and the backend resource to actually bring in all this pediatric cancer data from donors and CBTN, Kids First, and other resources like Target, to bring together all this data, harmonize the data, and bring it and deliver it to both clinicians, pharma companies, and anyone else who's interested in doing studies on this data. So we're also adding tools and data on a regular basis. So what is the data that's in there currently? So right now we have data from the open, as I mentioned, the open PBTA data from CBTN and PNOC. We have data from Kids First Cancer data sets, PPTC data, for Pediatric Preclinical Testing Consortium, the target data from the Target Project, and also we are bringing in reference data from the GTEx, because right now there aren't any current great normal tissue data sets for children, which I'll mention at the end of the talk. We've analyzed and harmonized that data together during different pipelines, and it's gone into this molecular targets database, and we're delivering the information on those relevant molecular targets from the FDA and their molecular profiles in our initial release, and we're growing the resource pretty much every day. So as a quick, how we do this is we have a primary alignment environment called Cavatica, which has come out of 7 Bridges, between a partnership between D3B and 7 Bridges at CHOP. I'm sorry, D3B is at CHOP and 7 Bridges as a company. But that partnership has come up with a great cloud-based resource for studying pediatric and data sharing and studying pediatric cancer and other diseases. And it's a great sharing platform where investigators can get together, share their data, and analyze it. The secondary analysis pipeline has come out of it's called Open PCAN, and it actually came out of the Childhood Cancer Data Lab, as one of the partners was Alex Lemonade Stand and CHOP. And Open PCAN was forked off of that and addressing a number of different cancers beyond brain cancer. So it's kind of a big umbrella of analyses. And it's Jolyn Rokita on the right-hand side there is one of the people who's managing that development. And if you're into that kind of thing, it's on GitHub if you'd like to look it up. So our secondary analysis at OpenPCAN, as I mentioned, oh, actually, there's a duplicate slide. Let me skip. Okay, so I'm going to show you a quick demonstration of this platform. You can go to molecular target ccci.cancer.gov, and you'll be delivered to this particular website. And you can start typing, either you can start typing in a disease of interest here, and you could be brought there, or if you'd like, you can go over here to the pediatric cancer data navigation, which is much more up to date with our recent data as we harmonize the platform over time. This is our first release. So if we were interested, in example, in MICN and medulloblastoma, you can search and go below the fold here, and you'll get information on you could click on the gene symbol itself, the disease itself, or the evidence page linking the disease and the gene. And it also shows you what kind of data is available. It doesn't tell you it's significant data, it just tells you it's available. Now, if you didn't want to just look at medulloblastoma but MICN across different tumors, you could scroll down, and of course here you're less filtered, but you can look at the evidence of MICN in any of these different tumors. But if I wanted to go back to medulloblastoma, I can click on that, hit search, and I can go to the evidence page. I'll just drop right into the evidence page. And here you can see that this is the evidence for MICN in medulloblastoma. Because we use the Open Targets platform, all these things that you see down here in blue that I'm highlighting are data that have been available from the original Open Targets platform. So they have a cancer biomarkers data on this, data from the Expression Atlas over in Europe, the Europe PMC, the PubMed Centrals for text mining. So there's lots of information on publications, ClinVar for somatic mutations, and Inogen for somatic mutations. This information came into us as we're introducing our other data, which has been a big boost for us. In addition to this, these in green here, and I apologize for people who might be red-green colorblind. We're going to work on the colors. But right now in green, we have the OpenPCAN gene expression and the OpenPCAN somatic alterations. If you click on this, you're brought down to the evidence page for gene expression. I know it's hard to read because it's probably very small, but for what you're looking at here is, and I'll go to log 10 so you can really see the difference. We're running a server that's delivering this data remotely. You see medulloblastoma is here, and this is the gene expression in medulloblastoma across a number of different patients, in contrast to normal tissues across the human body in adults, post-mortem data in adults. So everything from adipose tissue, here's a number of different brain regions in adults, and on and so forth from the GTEx project. So this is in particular the gene expression of MICN in medulloblastoma versus normal tissues. We also have information here on somatic alterations like single nucleotide variants, gene fusions, copy number variations, etc. And we have links out to other resources, including PhD Bio and some of these links. So you can look at if it's a fusion, you can look at any kind of information on it. And you can go to the information on the fusion and any information on its rate in the data set that we have. The other things that you can look at here is you can directly click into MICN, and you could be brought into the actual gene expression across numerous pediatric cancers at this point. So not only do you have gene expression in medulloblastoma, but you also have high expression in neuroblastoma and secondary tumors. So we could go to log 10, and then, if you're a clinician or someone who's interested in development, or you have a target that you're interested in besides MICN or MICN itself, you can ask whether or not this is a good target, not only versus normal tissues, but are there any other cancer data that has evidence that this is a gene with high expression. So you're able to look at MICN directly in these other tumors. And then you can also look at copy number variants in other tumors here as well. For example, neuroblastoma. And if you scroll over, there's link into PhD Bio if you'd like to look at the oncoprints of these across multiple patients. We've made sure that we put the same patient data in PhD Bio as is in this this particular platform. So it'll allow you to actually go back and forth between the two resources to look at more specific patient level data versus the overall data right now that we have in the cohort. And as time goes on, this will continue to develop into a more specific resource, but that's how we're solving the patient level data for now. So this is the molecular targets platform. If you're interested in the FDA's pediatric molecular target list, if you'd noticed I didn't point that out, but MICN had one of these R's next to it, so it means it's on this target list. So any gene that you click into, you'll be able to get information on pediatric cancers for any of these genes here, and you can get information if you want to go into a particular tumor type. You'll be able to look to see how it what the evidence is for that. We're also improving there's a lot of things that need to be improved in this platform, including how we're categorizing our histologies and making them more relevant for today's treatment strategies and how clinicians think about different types of tumors. So we're working with various organizations to make sure that gets in this platform. Right now, we're kind of blanking, like neuroblastoma. We don't have outcome specific types or molecular types for different tumors. Sometimes they're just under a blanket category right now, which may or may not be useful to some people. So we're working on improving that. And if you want to give us feedback, you can certainly contact me or any of the feedback links on these pages for information. And if you'd like to learn more about this. So I'm going to end with just mentioning the people that have been making this possible. It's been a productive partnership between the National Cancer Institute, Frederick National Laboratories, who's standing up the platform on the web, and all the people at CHOP that have been working on this project since it began about a year ago. And I'd like to have special thanks to the Wellcome Trust, the Open Targets platform team members over at the Wellcome Trust, and of course, the Childhood Cancer Data Lab at Alex Lemonade Stand for helping originally set up the post-secondary analyses of this entire project. And of course, there's a whole panoply of different people that have been involved in donating their time and energy. And at CHOP, there's a number of people that have been involved. John Maris and myself are the PIs. Adam Resnick, Sarah Tazian, and Sharon Diskin are the clinicians that have been clinician scientists that have been actively involved in the platform development. Of course, all the other people here on the list, including Joe Lynn, Alvin Farrell, and Asif Chinwala, so for their leadership. So thank you very much. And I want to do one more call out to the GTEx project, which many people may not know about. The DGTEx project is following up from GTEx. It is underway now. I'm one of the PIs out of, including Rebecca Lynn here at CHOP, where we're going to be trying to source normal pediatric postmortem tissues from the brain and other parts of the body, from age-matched children basically between newborn and age 18, which hopefully will help accelerate some of the ways we understand childhood cancer development in the future. Thank you, everyone.